So my name is Huiting Zhang, and as Julie has introduced, in the a few minute introduction, there are a lot of big words like transcriptomics, genomes. So I'm not gonna go too deep into it, but but I try to use some analogy to help you better understand what exactly these words means. But before that, I just want to say that we have heard a lot about wine chemistry and what are the compounds in the wine and all that. But for my talk, I can step back a little bit, but focus on the grape itself. Okay, so we begin with when there is smoke. How would you respond? So if you're an animal, you can run away, right? And if you are a human, you can run away while calling 911 and get someone to take care of it. However, if you're grape in a vineyard, you can't run away from it. You may hold your breath if it's a very transient smoke. But if it's a prolonged smoke exposure, especially when there is a wildfire nearby, you can't hold your breath forever. Even for plants, sometimes maybe it's possible to close the stomata for a certain period of time or reduce maybe. Yeah, we didn't really do any of those analysis, but maybe it's a possibility. But the theory is if the smoke is going on for days, it's unlikely that the plant can just shut off their respiratory system and not sequestering any of those compounds. So what they may be able to do is kick off their detoxification pathways, including I think many of you have talked about that conjugate some of these toxic compounds with sugars, makes them less toxic in the fruit itself. However, what this causes in the wine industry is that sometimes these compounds, they may be released during the fermentation process or some of the other processes during winemaking, but usually you can't guarantee that all these conjugated compound will be released during these processes. So what it end up happening is during the storage of your wine or even the tasting of wine, some of these compounds get released, and that will affect how the wine tastes or smells. So my research mainly is focused on to understand how grapes respond to the smoke at the big words, transcriptomic and the metabolomics level, with a goal to identify some key pathways and the compounds involved in this response. So what is transcriptomics? So here is a diagram that I just want to remind some of your biology classes that you take, maybe in high school or maybe in introductory biology classes. So when we talk about how, for example, a biological process happened, there are many different layers in terms of a regulation and all things working together, and it all starts from, usually it always starts from the DNA. And the DNA, it contains segments of it that will encode certain proteins. And then these proteins, they either build a part of the cell or they carry out some functions, for example, enzyme, to break something down or put some things together. However, between the genes and the proteins, there is another layer, which is the RNA. So the RNA, they are single-stranded molecules, not like a DNA, they're double-stranded. And then, so these single-stranded nucleic acid fragments, they are activated when there is a signal. So they get transcribed from the DNA. And then, so those little fragments then go through some machinery that eventually encodes a protein that carries out its function. And when we talk about a transcriptome, so what we are aiming to look at is to measure all these RNA molecules that a cell or a population of cells that produces. So that is an estimate of how different a protein group will be made after in different treatments or after a certain event. Okay, so this is what we are measuring. So we are aiming for the middle layer between the genes and the proteins. So these are RNAs. So just put it in an analogy. So this is the Spokane Symphony. And if you think of each player or each instrument, they are always there, right? Like at least during like one show. So these are the genes, for example. So they are always there. However, and then like the final like music that comes out of it is like the either the proteins or the metabolites that you ended up like receiving. Okay, the intermediate part of it is like who is playing at which time. So for example, the violins, maybe they do not play at the beginning, so they are like tuned down, but like in the middle, they started playing and they take over like the majority of the the symphony, like, and then at the end maybe they just tune down again. So that could make the change from a happy song maybe to a sad song. Like you are like at a very happy like symphony, and then the next song is like a Les Miserables. So you are okay, yeah. So this is like a very similar to how like us the genes in a cell the genes themselves don't change, but which genes get turned on and turned off really determines whether it's a happy song or a sad song. So in terms of like a grape or when grape are developing or when they are experiencing smoke stress, so what we observe from like the physiological or like a phenotypic perspective is like during normal growth, you can see the grapes are getting bigger. And then if you measure like some physiological changes, you can see like the cell wall is expanding, the cells are getting bigger, and they are maybe also producing more cells. And then there is accumulation of sugar and anthocyanin and also like aroma compounds. And when the grape is like experiencing smoke, some of these processes like anthocyanin development or ripening, they may like slow down or like these. So then because the cell is like just putting more energy into dealing with the stress. And then on the other hand, what you will see is like some stress signaling start to kick off, and then it will start like some detoxification pathways or like initiate some like a heat response like a pathways. But these are like what we can measure from the like a physiology or using some like a chemistry method. But what I'm interested in is like what are the gene responses that are different in these different conditions. So on the bottom, I'm showing like just two examples of like what potential gene expression changes you may like observe. So on the left, when it's during normal development, you may just see some genes like change their expression from low to high in like a very gradual like scenario. And then, but in terms of like a smoke response, the same group of gene, they may completely like change their expression pattern. And some other genes that were not a player during development, they may start to like increase their expression to respond to this smoke event. So the specific research questions that I'm asking like for this project is, first, how does the grape transcriptome like change over time after smoke exposure from the beginning to like when we stop the smoke and even after the smoke? And then the next question is like, which of these response genes are reproducibly detected across years and also developmental stages? And also including, as Tom mentioned, there are like a few switches in like what kind of like a palette we use. So maybe some of those genes, they are specifically responding to only one type of palette versus the others. And also what pathways and the candidate genes may underlie this response. Just a quick look at the setup. So instead of taking the grapes back to the lab, our smoke exposure experiment was done in the field in hoop houses. And so we have 3 control houses and 3 smoke houses. And the smoke is done in both pre-variation and post-variation grapes. So this is just a look at the inside of the house that Tom has already described. And I don't know if you can see it very clear on the screen, but like smoke pipes, they are embedded right underneath the canopy, so that to make sure like the grape can receive constant amount of smoke exposure like throughout the experiment and from the beginning of the roll to the end of the roll. And then we also have some like detectors as shown on the picture that captures like the smoke intensity level. So, and also the samples we collected, we did it at many different time points. So not just like we want to know after 24 hours what happened, but we want to know like what's happening during the smoking process from the beginning to the end. So at the beginning, we sampled these grapes every 2 hours. And once we reach like 12 hours, we kind of slow down and collected samples every 6 hours. Okay, so the red here shows like the time period that we are collecting the samples during smoke, and then we have a few samples after smoke. And then because the transcriptome, just like the symphony, if you like change the environment, if you like collect the sample now, and then like 2 hours later, it could be a totally different like response, because after you clip the grapes from the vine, then they may have some like wounding response or other response. So what we did in order to capture like a snapshot of the transcriptome at the moment we sampled, we flash frozen these samples in liquid nitrogen, so that just to freeze everything as quickly as possible. And we did these for a total of, so we repeated the same experiment for a total of 3 years at this moment. And yeah, hopefully we can like add another year of data to further strengthen our discoveries. So now that the berries are collected, the next step that we did is, so we took these berries, we fresh froze them, and then we isolated these RNA molecules from them. And if a gene is expressed at a high level, and from the RNA perspective, we will see many, many, many molecules of this same RNA in the sample. And then we send these for sequencing. So this is a method just to read which RNAs are like more abundant than the others. And once we get that RNA sequencing data, we analyze the data just to see like comparing the control and the smoke, which gene has a higher expression level in the smoked versus the control, or like has a lower abundance in the smoke versus the control. So the goal here is to identify which genes are changing at these time points during the smoke exposure. And I've been on this project for about like a little bit more than 2 years. So we do not have a very comprehensive analysis for everything yet. So I'm gonna just show like some preliminary result that we found from the data set that is generated. So first, the first analysis that we did is called differential gene expression. So we identified statistically which genes are expressed in the smoke samples, like either at a higher or lower level compared to the control sample. So we did it for each time point for each year. So these are all like a pairwise comparisons. And then from there, we also want to know like what's shared and what's unique at each time point or in each year. So we did a comparison like across different time points. So for example, all the samples from 2023 pre-variation, we want to know which genes are like always higher expressed than the others. Then we also, and we also want to see like across years, whether like if we change the pellet or maybe there are some other environmental differences, which genes are constantly consistently up or down regulated at a given time point across like different years. And we are also interested in to compare like a pre and a post variation at the same time, but this is still an ongoing analysis, so I won't be able to share too much about that today. But hopefully we can find something interesting, because I think a lot of you are thinking about like what if whether the smoke happened in like pre-variation versus post-variation, whether that will like cause a difference in the grape or in the wine. Okay. So this is so let's just first look at the structure of the data. And so here is showing a PCA plot, and the points that are closer together, they are more similar in terms of the symphony that the plant is playing. Okay, and then they are colored by time points. as you can see, the same color they usually group together, which means time, like when you pick the sample, is the main driver of which tune the cells are playing. But if you look closer to some of these samples, you do see separation between the smoke and the control samples. So I don't really know if I can point to them, but you just have to take my word for it. Yeah. So once we have this data, yeah, it makes sense because like at different times of day, your circadian clock is different, especially like if some samples are like during the day, some are during the night, there are definitely a lot going on than just the smoke. Okay, so that makes sense. So then the next question we ask is like, okay, now let's look at the genes that are differentially expressed across different time points. So first we looked at like for each experiment. So for example, this picture here is showing the differentially expressed gene from the 2024 pre-variation like samples, and we only look at the genes that are upregulated in the smoke sample compared to the control sample. And the y-axis on the bottom near the orange bars, these are the time points, and each dot represent like the category. So, for example, if you look at like the first few dots, so they're not connected to anything else, which means these genes are uniquely upregulated at a given time point. But if you see some points that has a line connecting to other points, which means these genes are upregulated in both all the time points that the line connects them to. So the first thing we noticed is that the most abundant differentially expressed genes, they're unique to each time point. But then which means the response, so we didn't see many genes that are shared across all time points. So this gives us it allows to make a hypothesis that the smoke response is likely a layered and time-dependent response, or maybe it's a dosage dependent response. So it's not like the grape is just doing one thing throughout the whole exposure period, but rather something got initiated, then later on it started some other response. So that, yeah, but we do see like the later time points, they share more of the same differentially expressed genes. So our hypothesis is although maybe across the grape started a little different, but at the end they are all doing the same things to survive. Okay. Right. Then we also wonder, so for those genes that are either turned on or turned down at different time points, are they reproducible across different years? So here is just an example of time point 12 that we looked at the overlapping genes that are turned up or down during pre or post variation, across different years. Although we see a lot different genes, as you can see, the tallest bars, the vertical bars, they are the genes that are not connected between the dots, meaning they're different. However, we do see quite a lot of genes that are shared between different years. And given that we have a lot of environmental differences and palette differences, so our hypothesis is that those genes that are shared across years are more likely the core responsive genes in this process. So here's just an example of the expression differences of those genes across 2 years. And on the top of this figure, you see a green bar and a cyan bar. This represents 2 different years. And beneath each of them, you see a red bar and then a brown bar. And the red is a control sample, and then the brown is a smoked sample. And the red color underneath it means higher expression of a given gene. So you do see this repeated pattern in our samples. And then the next step, what we did is okay, now we have a group genes, but what do they do? Which pathways are they involved in? So we digged further and conducted some functional enrichment analysis. So pretty much this is just to look at which genes are, so among all the genes that are up or down regulated, what is the function of those genes and which pathways they are involved in. So the example I'm showing here is still the same time point 12. And the key pathways that we found that those upregulated genes are enriched in includes like the glutathione transferase and the ethylene receptors. But we did not only look at them like in like one sample. If we go back to like this experimental design, so we look at the differential expressed genes at each time point and trying to figure out like during these different time points as the smoke gets stronger and stronger, how the response changes over time. So at the time point 2, what we saw that is mainly upregulated are involved in some sensing of the smoke and also some like a vacuum activity. It seems like they're trying to either import or export some metabolites or some compounds in and out the vacuum. And what interestingly, what we see downregulated is sugar metabolism related genes. It kind of feed back to our hypothesis that it like slows down like sugar production, but whether that also means like glycosylation is also slowed down, we don't know yet. We need to look a little closer into that. Then at time point 4, as the smoke continues, what we saw upregulated mainly involved in the ethylene pathways. So at this point, these hormones, they start to like really kick in, and our hypothesis is that they're gonna initiate a lot of the downstream reactions. Then later on, at around like a time point 6 and 8, the main signaling pathways that are upregulated are like HSP heat shock proteins, which is not surprising. They are involved in all different kind of like stress responses, and also ABA. So another stress-related hormone signaling pathway is also enriched. And then around like a time point 12, we start to see a lot of detoxification pathway-related genes. They start to like become really active. And then at those later time points, we still see these like detoxification pathway genes that are very active, but at the same time, we also see more downregulated genes that are related to like a cell wall and also like some other growth related processes. So yeah, this is mainly in the pre-variation grapes that we saw those differences. We haven't had the chance to look very closely into the post-variation grapes, and it will be interesting to see if they share the same pattern or they are doing something different. But one thing I do want to point out is that we also look at both across time and across year at pre-variation. And one of the very interesting genes that we found to be upregulated in the pre-variation sample across all those years and across all the time points is this one. So it's a pathogenesis-related BETV1 family protein. Anyone heard of this protein before? Because I hate it a lot. This is allergen. Yeah, and this is a protein that made me unable to eat peach and the cherry for the rest of my life. So there is a similar protein in peach or cherry, I should say that, but they have very similar structure. Although the name of it is like a pathogenesis related, but it's a regulator in a lot of stress response in plants. So this is in pre-variation. And this gene does not show up as differentially expressed in post-variation. So statistically, it's not like that different. However, if you just plot their expression level, so I forgot to mention the red is a control and the blue is a smoke. So if you just plot their expression at the post-variation samples, you can still see a trend of higher expression in the smoked sample compared to the control sample. So this is an interesting one that worth further experimentation to see how exactly is this gene involved in the smoke response, and whether it could serve as a potential marker for if the plant is experiencing smoke at a certain level. So with that, my take-home message today is from our initial analysis, we hypothesize that the smoke induces a dynamic time or dosage dependent transcriptomic response. So the symphony that the cells are playing, it's not like a constant help me. I'm like a suffocating tune, but it changes over the period of smoke exposure. Then a subset of those genes are reproducibly detected across different years, so suggesting there might be a core like smoke response program that involves like hormone signaling and detoxification and and some like other mechanisms. And the interesting like a BATW1 gene family protein, it could be maybe a candidate of like core smoke responsive genes. Yeah, because it's constantly upregulated in the pre-variation and has higher expression in the post-variation samples. But this is a very rich data set. We have, I lost count, maybe like 700 samples, and we are still generating more. So we're just scratching the surface of all the data that we have, and there is a lot more we can do. So for example, our next step is to construct a stress response gene activity network. So those genes, just like in a like an orchestra, like in a symphony, it's not like one violin is playing without coordinating with other instrument. So these genes they have a coordinated response to something. Okay, so what we want to do next is like build this network to understand how these genes are related, how like they regulate each other, and then so that the differential express genes tells us which genes are change strongly, but this network analysis may tell us like which pathway work together to like respond to this stress. And in addition to that, the plot I'm showing here is from a different but related experiment that we use transcriptomic data to predict a phenotype or to predict a physiological trait. And on the x-axis is the actual values. And then using transcriptomic data and the machine learning, we are able to like predict this phenotype or the trait data at a pretty high level. So yeah, the first one on the left is the training data. Then we tested the machine learning program like in some like different data, like different cultivar, different year, different. So this is apple data, so different orchard, and our machine learning algorithm was able to give like pretty good predictions. So like my plan is to like develop a similar method using the very rich like physiological data we have to build a similar model. So we can either use it to predict the level of smoke that the grapes are experienced, and if we can if we have like a pairing wine quality data, then eventually, hopefully, if we get like enough of this kind of data, hopefully we can also predict potential smoke taint risk just using the gene expression data that we gather from the grape in the vineyard. So with that, I would like to acknowledge all my collaborators, and I really appreciate Tom bring me into this very amazing project, and also the student who actually did all the analysis. So I was saying they are the artists, I'm just the art dealer. Okay? Right. With that, I'd be happy to take any question if there is